Okay, welcome everybody. This is the first BBK lecture of the new year, 2025, and we're delighted to welcome Professor Giles Budge from the University of Newcastle, who is not only a professor but also the Director of Research and Innovation in the School of Natural and Environmental Sciences. And some of you may know him from earlier work about yellow-legged Asian hornet and modelling its spread. But tonight we're delighted to have him talking about chronic bee paralysis virus, something that we're encountering more and more. So if you'd like to join in with the quiz coming up, there is a QR code. Scan the QR code and just hold on the introduction screen that it gives you, and then Giles will take it away. Over to you, Giles.
Brilliant. Thank you, Jane. Thanks for that introduction. And Happy New Year to everybody. And thank you for joining this evening to find out a little bit more about chronic bee paralysis. So confusingly, chronic bee paralysis is the disease, and chronic bee paralysis virus is the organism that causes the disease. So we'll run through that today as well. And yes, I'm hoping to run a short quiz to talk a little bit about chronic bee paralysis, to get some information from everybody on the call about whether they're confident with seeing the symptoms, recognizing the symptoms, perhaps if they've had it before, perhaps what damage it may have caused before. And the idea of that is that I'm going to share that back in a BBKA news article that I think I'm due to send in soon, within the next couple of weeks anyway. Okay, so I work at Newcastle University. I've been there for about 7 years, and I work within the modelling, evidence and policy group. And we're quite a diverse group of scientists with lots of different areas of specialism. But one of the things that we are able to do is investigate complex biological systems. And as we all know, trying to keep honeybees, they are certainly a complex biological system. So I'm delighted to be part of that group because it means that I get access to people who perhaps use methodology for looking at spatial or community ecology, and I can try and apply that to better understanding bee disease and bee disease spread. So it's a great place to be for the bee lab at Newcastle. Here you can see lots of different people who've been through the lab in recent years. They don't all work there now necessarily. And you can see some of the different organizations which have funded us over the years. And when I'm talking about the results today, some of the science that we've been doing, it's not science that I've been doing in isolation, it's science I've been doing as part of a team. And these are the people that have been helping with that science. I would be remiss of me to miss out our final members of the bee team, which are the bees. We've got 2 apiaries. We've got a research apiary up at Cockle Park Farm, just north of Newcastle. And we also have an apiary on the roof of one of the buildings. I'm hoping that when I go and see them next week, given the storms that are coming in, they're still on the roof of the building. Fingers crossed, fingers crossed. But yeah, it's really, really useful to have bees on site near the lab in Newcastle. Quick summary of the presentation. We're going to be talking a bit about quiz in a second, and then we're going to move on to run through what we know. And I'm just taking you through about half a dozen interesting publications from the literature that have looked at chronic bee paralysis that give us some clues about how the disease might be working, how it might be operating. And then this last part of the talk will be running through some of the progress that we've made up at Newcastle University to give us a better understanding of this disease. So here we go then. Let's give this a go. This is the code. So this is the QR code again. Hi everybody, this is just a little bit of bonus material really. I'm going to run through a short chronic bee paralysis quiz or survey that I did with those people attending the live lecture, and run through some information that helps us to understand a bit more about the beekeepers that joined us on the call, and also about their experiences with chronic bee paralysis. First up, we asked this question: how long have you kept honeybees? And we had nearly 120 responses, and you can see that the vast majority of people on the call have not been keeping bees for very long at all. Less than 5 years is probably the majority of respondents. If you look here over on the left side, you can see 0, and that just means that some people that were joining us didn't keep bees yet or weren't beekeepers. So here we have some more data that tells us a little bit about who was on the call. And the question was, how many honeybee colonies do you keep? We had around 130 responses, and you can see that actually the vast majority of people that were on the call either keep no colonies, or at least very few, or they keep, I think the mean was about 11 colonies overall. And then we've got a few what we might call professional beekeepers that come up here and keep more than 40 colonies. But in the main, we've got amateur beekeepers on the call. And that's also shown when we look at the number of apiaries. In the main, most people had a single apiary site for their bees, which is pretty typical across the UK. So here's another interesting question. I asked everybody, do you feel confident you can recognise chronic bee paralysis? And as you can see from this, around 2/3 of people on the call felt that they were comfortable that they could recognise this disease. And remember, this is before I'd been through the symptoms with them. Okay, so let's explore this question a little bit more and go back to the data that we saw earlier around how long people have kept bees. And if you remember, on the main, people in the main were amateur beekeepers that had not been keeping bees for too many years, certainly fewer than 10 years for most of the respondents. We can overlay information on this plot, which includes whether people felt confident they could or couldn't spot chronic bee paralysis. And when we do that, it's fascinating actually, because the more experience you have, the more likely it is that you feel confident that you can recognise chronic bee paralysis. And in epidemiology, this creates a bit of a conundrum that we call ascertainment bias, and that means there are certain cohorts within the beekeeping population, those with little less experience, who are less likely to report the disease if they see it out and about, because they might feel less confident being able to recognise that disease. We then moved on to ask this question: have your bees ever suffered from chronic bee paralysis? And around half of the people on the call said that they felt that their bees had suffered from chronic bee paralysis. I don't know how much we can say from these data, because effectively we probably self-selected individuals that were interested in the disease because they felt that their bees had suffered from it previously. What we can do is, not surprisingly, show again, looking at experience, how long people have kept bees, that people felt they were more likely to have suffered when they can actually spot the symptoms. So after a few years of beekeeping, you can see that blue creeping in, where people feel sure that their bees have actually suffered from chronic bee paralysis. So the next question that we asked was approximately what percentage of your honeybee colonies suffered from chronic bee paralysis. And for this question, we again had around 100 respondents, and we were able to calculate from those respondents, if you like, a pool of healthy colonies, so those people whose colonies had never suffered, and also those people for which a percentage of their colonies hadn't suffered. So we had healthy colonies and we also had disease colonies. And when we looked at those data, it was around that 130 or so colonies were diseased out of 930, which gives us around 14% of the total colonies surveyed by the people that were attending the evening lecture had apparently suffered from chronic bee paralysis. So the next question that we asked was, what percentage of those colonies that were diseased failed as a result of the disease? And again, around 100 or so respondents for this question. So of the 130 colonies which were found to have disease through our small survey of our attendees of the evening lecture, around half of those, 62 colonies, were suggested to have failed. And that gives us something which is quite similar actually to the literature, which is between 40 and 50% of colonies that actually suffer from this disease fail as a result of it. So I hope you found that summary of our chronic bee paralysis survey or quiz from the evening of the lecture useful. I'll now return you back to the live lecture. We're going to switch back to the presentation now, and we're going to start going through and look at some of these symptoms. Okay, so in the literature, you'll see that you have different types of symptoms for chronic bee paralysis. You've got type 1 symptoms, which are trembling, shivering, shaking, and often these bees are displaced from where they would normally be. They often don't respond to smoke, and they will effectively cluster in places where you don't want them as a beekeeper. They'll stay on top bars, they'll go on lugs, and you can often end up squashing them because they don't move with the smoke. So I'm hoping that the video comes through for you of these bees all sort of shivering and shimmering on these top bars of this colony here, and lots of repeated movements, lots of moving forwards, moving backwards, not like you would normally expect honeybees to behave. So they're the type 1 symptoms, and what we've found is that these symptoms often occur early in the disease cycle. Type 2 symptoms are very different. Effectively, what you have are bees which can sometimes appear darker. They can be hairless and shiny and greasy looking. They can have nibbled wings because effectively sometimes the hairs have been removed by their sisters trying to remove these slightly sick bees from the colony. So you can have these bees that sometimes look like robber bees. They're very old in appearance. Okay, so these are type 2 symptoms. And in our experience, watching disease progress in the field, these symptoms tend to occur later in the disease cycle. Okay, so if you're seeing these bees, they're nice and shiny, it probably means that they've that the disease has been there a little bit longer. Okay. A feature of chronic bee paralysis very much is the large number of dead bees that you can have at the front of the colony. So any of those bees that you saw shaking earlier, they would probably be dead within sort of 5 to 7 days of during sort of showing those symptoms. They won't live much longer than 5 or 7 days, and then they'll get thrown out the front of the colony. So you can get these mass piles of dead bees out the front of the colony. When I say dead, I don't really mean dead, because yes, the base layer of this pile that we can see here is very much dead and decaying. You can see a difference in color here. But if I look at these bees here, you can see that they're a different color. They're clustering. They're actually still alive. And if I was to blow perhaps on some of these bees here, the next layer down, they might twitch. They might not actually be dead. They're actually paralyzed because the virus that causes this problem is attracted effectively to some of the sort of neural tissue, and it can cause some degradation in that neural tissue, and it can lead to paralysis. Okay, so dead bees aren't always dead. So part of dead bees can sometimes be twitchy, and there can sometimes be some symptomatic bees that have been thrown out the colony on top of that pile of dead bees. This is a beekeeper from Yorkshire, and I think it's a really nice example of the dead bees on the bottom board. You can often get piles on the bottom board. The bees can't keep up with throwing out the dead bees and the dying bees. But when you look at these, you can see actually some of them are actually still twitching. They're not actually dead, they're still moving slightly. And if you were to blow on some of these ones that appear to be stationary, you might get a little sort of tarsal twitch or a twitch of the leg or something, because they're actually still alive. And I'm going to show you what these virus particles look like. So this is chronic bee paralysis virus if you had a very powerful electron microscope. And effectively what we're seeing here is the causative agent as was identified by Bailey in 1963. So these are very unusual virus particles because they're anisommetric. It means that they're asymmetrical. So you can see that if we start to go around the outside of one particle and then another particle over here, it's a different size and different shape. It's a very unusual virus. It doesn't really have any closely related viruses at all, actually, that we know of. But this is the virus that's causing the problems. And this virus is very much attracted to and replicates really well in neural tissues. So how does this virus move around? Well, the first paper I wanted to point you towards was one by Magali Ribière in 2010. It's an open access article and it's got lots of information on chronic bee paralysis, including the modes of transmission. So what we mean by this is how the virus moves from bee to bee. Okay. We know that the virus can spread between adult bees using fecal oral transmission. So if you have feces from a bee which is actually suffering from the disease, those feces can actually transmit virus particles through into honeybees and cause disease in those honeybees. So that's the fecal oral route. You can also have mechanical, and mechanical just means that the bees can rub up against each other and can end up passing the virus between themselves, okay? You can have transovarial through the queen. That means that the queen can become infected and then it can lay eggs which contain chronic bee paralysis virus. Now, what I would say about this is this refers to one or two papers from around 20 or so years ago where they had very, very sensitive techniques to detect those virus particles. And it was never really shown that those individual eggs ended up developing into adults that had the disease. So it was really a trace level of virus, but nevertheless, it does go down as a transovarial transmission from the point of view that the virus moved and passed from the queen through to the eggs. Then, of course, injection. So like many viruses in honeybees, if you inject them directly into the hemolymph, they can get going, they can cause quite a rapid infection, and they can lead to the death of the bees very, very quickly. And you might say, because you can inject the virus into the hemolymph, you might ask the question, perhaps Varroa then, as it's moving around, is moving the virus around and causing the problem. Actually, we don't think that's the case. We don't think that Varroa is a significant vector of chronic bee paralysis virus. Okay, there are a few publications which talk about correlation, but actually overall, it's quite weak evidence compared to the number of times people have tried to actually move the virus around with Varroa mites and failed. So that was a relatively recent paper in that it was around 15 years old. Now we're going to go right back nearly 100 years ago to a brilliant paper by Burnside. And he was working in North America, and somebody brought in some colonies that were suffering from chronic bee paralysis. And actually one of the problems with working on this disease is finding colonies with the disease to experiment on. So when somebody brought him some colonies which had the disease, he thought he'd experiment, and he did some lovely experiments on the disease. The colonies that were struggling were headed by sick Italian queens. Effectively, the queens were okay, sorry, but the colonies were sick. Okay. And the first thing that they did was to move over pollen and honey, whole frames of them, into colonies that were headed by carniolan queens that actually were local and didn't have any disease that was being shown. And they wanted to see whether or not, having moved over that source of inoculum, would those colonies that were healthy start to show disease symptoms. Interestingly, no disease was found. Now they moved brood over to more healthy colonies. and you would expect, perhaps if this was foulbrood, that the colonies over here would develop European foulbrood, let's say, and the colonies over here would develop European foulbrood. But with this particular disease, no disease develops. So you can move whole frames of pollen and honey and whole frames of brood, and the disease didn't move with those sources of inoculum. The brood that was moved out of the Carniolan colonies came back into some of the sick Italian colonies in order to make up the numbers more than anything. And what was fascinating there is that disease in the adults, as they emerged, the brood emerged that was Carniolan. You could see that both the adults from the Italian queen, the Ligurian queen, and the Carniolan offspring both showed disease symptoms. So effectively, what Burnside was the first to show was that this is a disease of adult bees. So once this disease gets into adult bees, it can be passed between them, but you can move whole chunks of the colony around and you're not necessarily going to move the disease. Okay, so it's a very interesting old paper that gives us some clues about how this disease is operating. This is a paper by Bailey, fantastic paper from 1983, where he was really interested in trying to better understand whether or not crowding, increasing the density of bees in colonies, might lead to an increase in the disease transmission. So he set up some pots and he had some healthy bees and he had some diseased bees in those pots, and he wanted to see whether or not the disease would move at different densities. So that's the low density on the left and the high density on the right. Same size box, just more bees. Okay, so they're more crowded. What he found was that actually a few bees developed chronic bee paralysis. Okay, that should say chronic bee paralysis rather than chronic bee paralysis virus, so they did check for that as well. Okay, so about 32% of those healthy bees in the low density pot developed the disease, whereas 84% of those healthy bees develop the disease in the high density pots. This is some good evidence that actually perhaps the disease is more likely to move through healthy bees from diseased bees when the colonies are crowded. Okay. The next one I think is a really interesting paper. What you can see here is a typical winter on one of my colonies, because I always get these little black ants that make a colony on the top. I always have a bag of candy on the top of my colonies, just in case the bees need it as they go through the winter, and I can peep through and see how they're doing with eating it and replace it as necessary. But this is the warmest part of the colony, just above the center, wherever they might be. Okay, and we always get an ant's nest there. And this paper, there's a few papers that have looked at alternate hosts. I think sometimes honeybees get a bit of a bad rep because there are lots of different viruses that are associated with honeybees. But actually, some of the viruses that are associated with honeybees are not honeybee viruses. They're actually more accurately described as insect viruses that were originally found in honeybees. And what's interesting about chronic bee paralysis virus is if you do look at things like ants, and this is a couple of papers here you can see, if you look at ants that are raiding honeybee colonies with chronic bee paralysis, then actually you can find an awful lot of virus in these ants. The negative strand comment is important because that suggests that that's replicating virus that's been found within the ants. And what's interesting, what's even more interesting, is that the amount of virus that was found in those ants was 1,000 times higher than you had in dead bees that were suffering or had suffered from chronic bee paralysis within those colonies. And that suggests that the virus itself is able to replicate to very high levels in the ants. Okay. It's also been detected in commercial bumblebees. And I've got a really nice paper here, which shows some additional information. I think this one's also open access, so you can have a look at this one yourselves if you like. But the table that I'd direct you to is this one, which goes to look at how many samples have tested positive for chronic bee paralysis virus from all these different pollinators. So you've got these sort of sweat bees at the top here, and a lot of those test positive for the virus. You've even got some flies, these Diptera here, and a lot of those test positive for the virus. And again, what's interesting is when you compare these results in the same paper to Apis mellifera, actually 32 different species had more chronic bee paralysis virus in them than the honeybee samples. Okay, even the flies. So I think it's safe to say that actually when we're thinking about this as a virus, it's actually a virus that's able to replicate in other species that aren't honeybee, and you can often find it in lots of other species too. And the last paper I wanted to point you to is a paper that we published a few years ago, which used information from BeeBase to try to answer this question, really around whether or not the levels of chronic bee paralysis virus were increasing in England and Wales. Okay. And you can see here that if we look over time and we classify cases of chronic bee paralysis, then you can see that over time we see an increase in those cases. And this is effectively up at about 3%. When we started the study, it was down at about sort of very, very low, below 0.5%. Okay. And then you can see on the right side where these clusters were in the landscape. So you can see that this disease is being recorded more by the bee unit over this time frame. Actually, I've got some more recent data than there's in this paper, and it kind of continues to increase for a couple more years, and then it sort of tails off and stays at a similar level at around sort of 2.5 to 3% in more recent years. Okay, so it's definitely increasing in recent years. And if you look back through the history books, you can actually see that this disease has been going up and down quite a bit in the past. And there are papers that look back in the 1960s that suggest a similar sort of increase over a period of time. And so it's not the first time that perhaps this disease has emerged, which is why I've put up there, it's re-emerging. Okay. So we've got some knowledge gaps, right? There's lots of things we don't know about this virus and this disease. And we don't know how common it is. We don't know whether the disease just seems to appear. I don't know if that's a lot of people's experience on the those of you who've seen the disease in your bees. It wasn't there one week and the next week it was there. It's really unusual for a disease. Does it really happen? What are the disease risk factors? What are the things that might be going on which might be increasing the risk of our bees? Are some ages of bees more susceptible to perhaps oral infection with chronic bee paralysis virus? Can pollen help prevent disease? Are dead bees a source of infection? Are drones a source of infection? And which control options work? And I'm going to try and get through some of those things now. Okay. So let's have a look at some progress to try to address some of those evidence gaps for this particular disease. First question was how common is the virus? And if we look at some previous work that's been done in France, we can extrapolate to say that actually the virus itself is present in about a third of apiaries, and the disease is running at about 1.5%. So the virus in France is much more common than the disease. So more often than not, when the virus is present, it doesn't actually cause the disease. Okay. What about England and Wales? Actually, these data are England, Wales, and Scotland. It's actually a little bit higher up than where I've cut off the map there. So apologies if you're calling in from Scotland. Okay, what did we find in this paper? What we found was it's quite a small sample size, but interestingly, we found some very, very similar numbers to France. Around a third, again, of all colonies now, rather than apiaries, tested positive for chronic bee paralysis virus, but the actual virus itself was present in around 1% of colonies. So we did this work a few years ago, and if you'll remember from my plots just now, it continues to rise up and rise up after the point at which we sampled here. So I think this was actually done in about 2014, 2015. Okay. So again, the virus is much more common than the disease. We can continue to think about the virus in different ways, because we can detect the presence of the virus, but just because we detect the presence of the virus in asymptomatic individuals or asymptomatic colonies, it doesn't mean that there's the same amount of virus that's present in the symptomatic colonies. And what this plot's showing is the log amount of chronic bee paralysis virus here. Okay, so as we go up here, we get more and more and more, but it's log scale, remember. And we can see actually that our symptomatic colonies have got an awful lot more virus in them than our asymptomatic colonies, even though some of our asymptomatic colonies tested positive for the virus. In fact, it's 230,000 times more virus, okay, in those symptomatic colonies. So something's happening whereby you go from having a little bit of virus in some of these asymptomatic colonies to having an awful lot of virus in other colonies that develop disease. Okay. Okay, so the next question is, does the disease just appear? That's certainly the experience of most beekeepers. They feel that suddenly something's happened. At one inspection, everything's fine. At the next inspection, there are thousands of dead bees out the front of the colony. So this is an experiment that I did with a postdoc at the time, Philippa Holder, and we worked with the bee farmers and Arnia, and we went around and we set up 3 different apiaries. So we were looking at colonies and monitoring colonies that were in bee farmers' apiaries for which they often had chronic bee paralysis. So it's kind of a bit of a guess. So our best guess is if we go and we set up and we start sampling earlier in the year and we start monitoring symptoms, maybe we'll see some disease in some of these colonies. Okay, we had a site in southeast England, one in the Midlands, and also one in northwest England. Okay, and we went back and we monitored these sites every 2 weeks to try to better understand the disease dynamics. What did we find? Well, it was a 600-mile round trip for us. It took us two or three full days in order to do each one, and by the time we got to here, and I dragged poor Philippa around this trip 5 times, and we were finding no disease in any of these colonies. You can see we've got 5 colonies here at site 1, 5 colonies at site 2, and 5 colonies at site 3. And we've done 5 visits and we haven't found any disease. So we're getting a little bit fed up with going, shall we stop the experiment or should we carry on? Well, we're glad we carried on, because then we started to see things. Okay. We had these dead bee traps outside the colonies to try to capture any increased drop of adult bees from the colonies. And what we started noticing on this 6th visit was that actually there were more dead bees in the trap than there were before. Okay, so we saw this increase in the trap first, and actually we saw it, you can see, in all of the colonies that then developed symptoms. The S and the red represent symptoms of chronic bee paralysis at that colony level. Okay, so it did develop in 6 colonies. It didn't develop in this apiary over here, although we did start to see some increase in the trap. It never really got into the colony that we could see. So what did we learn about this experiment? We learned that the trap told us, and we tested these, and they did actually die of chronic bee paralysis virus. The trap had an increase in dead bees here. 2 weeks later, as researchers with a really highly sort of honed ability to spot the disease, we spotted it here, but the beekeeper said he wouldn't have spotted it until it was here. Okay, so that's actually a little bit later, because we were really looking for it, and normally you're not looking for it necessarily. Okay, so consistently we were finding that the beekeepers, actually these are bee farmers, they're very experienced beekeepers, they really know their stuff, they really know and understand this disease, but we were spotting it a couple of weeks before them, and the trap was spotting it a month before them. Okay, 4 weeks before them. But if we go back and look at some bees that we collected here, right back in the 2nd visit back in March, in some of these colonies, we're finding virus in dead bees, very high levels of virus in dead bees. That tells us actually that it doesn't just appear. This is like many epidemic diseases. It has to go through cycles and it takes time to build up. And perhaps we only notice it when a certain number of bees are suffering from that disease. Okay, so it doesn't just appear. It's there and it's burning away and it's chipping away slowly, and then we notice it as it really ramps up, as the epidemic really takes off. Okay. So one of the things that we answered in our earlier paper was what the risk factors might be. And I just wanted to remind you, really, you may have seen the paper before, but I wanted to remind you about what some of those risk factors were. So these are things effectively, this is like classical epidemiology. You're looking at some data and you're trying to figure out what might be going on. So you're looking for some signal in the noise to try to help you to understand what might be going on. First of all, not surprisingly, when you've got that kind of increase that we were seeing, then it's more likely that you find disease the later in time you look. Okay, so that's like an increase risk. You can see here 1.25 means 1.25 times the risk per year. So it's increasing quite a lot each year over the period of that particular study, which went from 2006 to 2017. If you're a professional beekeeper, now we didn't actually know who defined themselves as a professional beekeeper, so we just said anybody who had 40 or more colonies was a professional, and anybody who had 39 or less or fewer colonies would have been an amateur. But if you're a professional beekeeper, you're 1.5 times more likely to get the disease, which is interesting, right? And we also found that if a beekeeper had imported bees within the last 2 years, then there was a 1.8 times greater risk of them having the disease. And there was quite a lot of noise around at this time about, oh, how imports must be bad and all those kinds of things. And that's not necessarily the case, because it could be that our native bees are just as susceptible as our imported bees. And in fact, a bee farmers survey from 2017 surveyed the members of the bee farmers and asked the question, have you had chronic bee? How much chronic bee have you had? Similar sorts of questions to what I asked you earlier. But they also asked the question, what sort of bees you keep? And you can see here that actually almost all types of bee that you might be familiar with, major imports, and also Apius mellifera mellifera or mongrels or local stocks, they will all have an opportunity to develop the disease. So it's not one particular type that gets it. We did get a slight increase in the signal for buckfast, but buckfast aren't the only ones, the only bees that actually catch or can show symptoms of chronic bee paralysis. So we wanted to try to investigate the small signal we had with the buckfast bees. And so this is Nicola Simcock, and she's a postdoc in the lab after Philippa. And one small experiment that she ran, and we're kind of stymied a little bit by COVID and things here, but she went to 5 different importers that were importing Danish queens and tested 32 different colonies and 32 different queens. And this is what she did. She went and looked at the colonies here where the queen was going to be put in within the next week or so, okay, and sampled the bees to see if we could find virus in these colonies before the queen got there. And she also sampled all of the attending workers that had travelled with that queen over from Denmark, okay, and looked for virus in these samples. The idea being that if this was clear, if the colony is clear, but the attending workers are smattered in virus, it might actually give us an idea where the risk is coming from. If it's the other way around, it tells us a little something else about the system. Okay. So what did she find? Well, actually, she found that all of the apiaries that were sampled had at least one colony that tested positive with chronic bee paralysis virus before the queens were inserted. And in none of the attending workers did we detect any chronic bee paralysis virus. Okay, so that tells us that there isn't an import risk per se for the virus, at least not in this experiment. But actually, the bees are being moved to colonies that already probably contain the virus. The virus is more common than people realize. As I mentioned earlier, about 1/3 of all colonies that we sampled actually have the virus at some level. Moving on then, let's have a look at some more experiments. So here we've got Theodora and Nicola again. And what we're interested in these experiments was to try to better understand how the virus moves into honeybees and causes disease when you actually expose the bee with an oral dose of the virus. Okay, so here's Theodora. This is a bee. Okay, and this bee is being held by her wings, and in this pipette we have a sugar syrup solution, and in that sugar syrup solution, in some of the bees we had no virus, and in other syrups we had some chronic bee paralysis virus. And the idea was that we were going to set up some experiments to see what age bees are susceptible. Is it as they come out and they close? Is it after a few hours that they're still susceptible, or do they become resistant? Okay, we're investigating again the epidemiology of the disease in the colonies. So we've got 3 treatments here with chronic bee paralysis virus that have been added. We've got freshly closed bees here, bees that are 3 to 7 hours old, and bees that are 24 hours old. In other experiments, we also looked at 48 hours as well. Okay, they're put in little cages in pots, and they're compared to those that are fed same ages, but fed effectively a very weak salt solution, okay, like saline. And when we monitor we basically monitor whether or not the bees go down with the disease. This is one of the bees that was exposed. I don't know if it'll work. Yes, it will. But you can see that she's showing very clear signs of chronic bee paralysis. She's got repeated movements. She's got some tongue lolling. Quite often with bees, as they develop paralysis, it can be one side, so they walk in circles because only 3 of their legs are actually working properly. So we saw some really nice clinical symptoms. We confirmed the presence of the virus as well. What did we find? So we found, if we look at the proportion of bees that are alive here, we found that just after a few days after exposure, 2 groups dipped down that were exposed to the virus and another group stayed high. Okay, so this is showing that these 2 groups are actually, we found out later that these died of the virus. Okay, a proportion of them died of the virus, whereas these ones didn't, that stayed at the top. And the difference is that the ones at the top here are either the controls or the 24-hour-old bees. So when they're 24 hours old, they don't actually die of the disease. When they're exposed when they're younger, both the groups showed a very similar pattern. And this is just one example from one experiment, but this experiment was repeated lots of different times and gave the same answer, that we just could not get the virus into bees that are either 24 or 48 hours old. Okay, it's actually the very young bees that are susceptible to oral infection with this virus. There we go. Yeah, young bees are susceptible to oral infection. Older bees are not. Next experiment that Theodora did was to look at pollen and wanted to ask the question, can pollen help to prevent disease? So she took some effectively bee bread, some pollen frames from a colony, and she made these lovely little pollen capsules, okay, made of bee bread, and put those in with bees, and put these in with some bees and not with other bees. Okay, we then exposed those bees to oral inoculation, same as before, same here, or inoculation, and we watched them to see whether or not the ones without pollen survived as well as the ones with pollen. Okay, what we found is within 10 days in this particular experiment, all of these bees at the top were dead, but only 50% of the ones at the bottom were dead. So it seemed that the pollen was giving some sort of tolerance to the bees, or the absence of pollen was making them more susceptible. And this is interesting because it's kind of in line with a paper that came out a few years ago by Dubois et al., where what they did here, which was really interesting, is they took some colonies which had chronic bee paralysis. They'd recovered from chronic bee paralysis. They then put some pollen traps on those same colonies, and the symptoms came back. Okay, and they weren't sure if it was to do with the abrasion of the pollen trap, whether it might have been a lack of pollen. And our results suggest that actually, if you've got low pollen, there's a chance actually that you are going to increase the susceptibility of those bees to chronic bee paralysis virus. Okay, yes, a lack of pollen increases susceptibility. The next experiments that we looked at were trying to better understand these dead bees and whether or not they might represent an important source of infection. And this is some work that Megan did, Megan Hardingham, who was a master's student with us for a summer. And very similar setup. It's pot experiments. In this case, what we have is these are supposed to be dead bees, in case the little crosses across the eyes. These are dead bees which have been exposed to chronic bee paralysis virus and have died of the virus. Okay, and these are healthy bees. And so you put these bees in a pot and you say, right, there's some dead bees in there. These ones are going to rub past these ones. Is it possible that the virus moves from these into the healthies? And then as the control on this one, we had some dead bees again, but these dead bees were healthy bees. Look, they're grey, they're not pink. Okay, so that's our comparator in this experiment. So what did Megan find? In her experiments, it was very, very clear, actually. If you add corpses of healthy bees, the bees live a kind of normal life when they're being fed sucrose, okay? You got a few dead bees coming in about 3 weeks. The dead bees are here, pinky purple, and the asymptomatic bees, the ones that are still alive, are red. What happens if you put corpses in which have effectively died of chronic bee paralysis virus? You get a very, very different picture. You get very, very rapid transmission and rapid death that continues in the pot effectively. So within, I think on here, look, within just over 2 weeks, all of the bees are dead. Whereas over here, just over 2 weeks, 2.5 weeks here, actually very few of them are dead. So really, really big difference between these pots, suggesting that bees that die of chronic bee paralysis are a very clear infection risk. And we wanted to quantify that. Okay. So this is an experiment I've not shared with anybody yet, but this is my apiary. I had chronic bee paralysis in this apiary in 2022. It all cleared up by about June 2022, something like that. And then I was pottering around and I realized that in the deep grass, I could see some of the residual dead bees. They'd broken down, but I could still find bits of cuticle, bits of bee, and I wondered if it was still infectious or would test positive for the virus. So I went scratching around, picking up these old dead bees, along with some leaf litter. And about a year after I'd last seen symptoms of chronic bee paralysis in the apiary, and I made some inoculum, which I either fed fecal orally, or I put into this is actually, I think this experiment was actually run by Megan again. So Megan then put in some of the detritus in with some healthy bees to see if they went down with chronic bee paralysis, a bit like we'd seen in the previous experiment with the dead bees, or I injected. Okay, or I think it's actually me doing the injections here. Okay, so we either injected some of the old dead bees that we'd found effectively in the grass to see whether or not we could cause chronic bee paralysis in these bees. And what did we find? Well, for a start, we could still detect the virus in the inoculum, which is good. If we fed that inoculum, then we couldn't induce any symptoms in the bees. If we put the inoculum in with bees, then some of them did die of chronic bee paralysis, so 3 out of 60, so it's a low level. And if we injected a few more, actually went down with the disease. But what that tells us is that many, many months after an outbreak, even in the old detritus that would have been a pile of dead bees months before, we still have an infection risk. Okay. And that's what's important, I think, about this particular experiment. We have to think about what we're doing with those dead bees if we want to manage the disease. I think this is the last one I wanted to mention. Our drain's a source of infection. This is our experiment. You'll recognize it from earlier. These are all of our different visits. I just wanted to say that we found virus in dead worker bees here, but we found virus in live drones here just 2 weeks later. Okay, so the drones had virus in, quite a lot of virus in. We also found drones with symptoms that we collected. Okay, you've got a worker on the left and drones on the right. Okay, so they do get symptoms, they carry very high levels of virus. And just for fun, we thought we would see whether or not they flew. So we got some workers with either early or late stage symptoms of chronic bee paralysis. And we threw them in the air to see if they would fly and orientate back to the colony. And you can see around 75% of those with early workers, early symptoms did, and about half of those with these late stage symptoms, this is like the type 2 symptoms, were still able to fly and orientate. When it came to the drones, we couldn't find a drone, no matter how sick it looked, that wasn't able, when we threw it up in the air, to basically orientate and go back to a colony. Okay, so it's interesting. They carry a lot of virus. They can become symptomatic. They can still fly, and they can carry very high levels of virus. So I think it's fair to say that drones are a potential source of infection for chronic bee paralysis. Okay, so control options, I'll whiz through these. I'm not going to mention this slide, but queen replacement, I've got no evidence that that works. This idea of doing a shook swarm, I've got no evidence that that works. Practical suggestions for control. This work from Bailey suggests that you avoid congestion. You don't want your colonies too congested. I think that's still valid. The work with the bee corpses that we were doing with the dead bees, wider entrances help those bees maybe to take those dead bees out. Remove the piles of dead bees, take them away from where they might actually be causing reinfection in the colony. So manage those dead bees rather than letting them rot down like I did. Maybe when it comes to pollen, good supply of pollen might mean that the bees are less susceptible to the virus. And this is the last one, really. The really interesting result that Theodora found was this idea that young bees are particularly susceptible. And so we were playing with this idea of comparing no treatment with colonies with brood separation treatment. So with the brood separation treatment, effectively what you're doing is you're taking the young bees, the brood and the queen upstairs, you're putting a split board in here with a new entrance in the opposite direction. You're leaving a super or something down here for the old bees that are going to fly out and fly in and come back to that bottom box through this normal floor. And we wondered if that would help to break the disease cycle by separating out those old bees that are carrying the virus from those young bees that are very susceptible to getting the virus. So the last result I wanted to share with you was some a small scale study that we ran. It's too small to say much, but it's a start, and it's something that I'd quite like to continue to look at. And that is if we compare no treatment, so we do nothing. These are colonies that all have chronic bee paralysis, with the brood separation method that I just mentioned, with this common advice that comes out, which is remove the floor so the dead bees drop out the bottom. If we compare those, when it comes to mortality, then the no treatment, about half of them die. As I mentioned earlier, that's quite common in the literature. The brood separation, the fewer died, but not significantly so. And the floor removal, more of the colonies died, and significantly so compared to the no treatment. But these are very, very small numbers. Okay, so we really need to try and get a better idea of what might be going on by trying some of these things out and getting more evidence to help to support any of these potential curative treatments. Potentially some future work there. All of the information that I've given you today is in a beekeeping information leaflet that's tucked away on BeeBase. I've given you the QR code again to find it, so that'll take you straight there, or the link is here if the QR code doesn't work on your camera. But all of the information for beekeepers that I've talked about today is in there somewhere. And this is just a summary to say it is an emerging problem as a disease. All types of honeybees are susceptible. Dead bees are highly infectious, so we have to do something with those corpses because they remain infectious for quite some time. Young bees are very susceptible, and absence of pollen increases that susceptibility. And we really need to concentrate a little bit more on gathering good evidence on what might be representative, preventative, and curative treatments. So thanks to everybody who contributed from the lab. We have a little pot that we can try and get people to put some money in to help us with the bee research, which does help us out with some of these small scale experiments. So if you want to put anything in there, that's the link. You can come back to it with a recording, I guess. Thank you for your attention. I'm very happy to take questions.
Thank you very much, Giles. That's really interesting. Can I ask a question? Early on, you talked about type 1 and type 2. And as you said, in the literature, we've always been told that these are different forms of the virus. Are you suggesting that they might just be different stages of the disease?
Yes, there's no sequence difference between those individuals, so there's no genetic difference in the virus that's causing those problems between type 1 and type 2. Okay, so if we sequence the virus from an individual showing type 1 or type 2, it's actually almost clonal in the same outbreak. And it's just something that we noticed when we kept going back to these colonies that had developed symptoms, and it's not just the ones in the study I showed you. We've done this quite a lot with other beekeepers over the last few years, and we've gone back and looked at their colonies over time. I looked at my own as well. And you don't really see those super shiny nibbled bees very early. You tend to see shaking bees first that appear to be, if you'd taken a picture of them and as a freeze frame, they would look healthy, but it's just their behavior that's different. And then later on, you see these where if you took a picture of them, they wouldn't look healthy. If it was a still picture of a shiny bee with nibbled wings, it doesn't look healthy. So that's what we found anyway.
Okay, thank you. Now we've got some questions in the chat. People were asking some practical questions about if you've had a colony with chronic bee, and you want to, what's the protocol for cleaning up, as it were? Does the hive need flaming or sterilizing? Obviously not the polyhives. And
Yeah.
what sort of hygiene needs to pertain to chronic bee?
Yeah, it's interesting, isn't it? Because I've just shown that the virus itself is very commonly found. But then you get these high levels of virus in colonies that are showing disease symptoms. Certainly in the bees that are showing symptoms. If you look back at the literature, and I mentioned it earlier, you can take whole frames from a colony that has showing symptoms of the disease and put it in another colony and they won't show symptoms of the disease. But equally, we took some pollen from a colony that had disease, and we took some pollen from a colony that didn't have disease. The pollen did test very weakly positive for chronic bee for chronic bee paralysis virus from the colony that had chronic bee paralysis. And we fed that pollen to bees that were susceptible, the young ones, and they didn't show any symptoms and they didn't catch the virus effectively. They didn't have any replicating virus within them. So when it comes to tidying up, I think you've got to concentrate your efforts on where the highest levels of virus are going to be, and that's going to be in the dead bees or the bees that had symptoms. I can only tell you what I did with mine, and that is I used all the same equipment. I did nothing to change anything about the equipment that I had. It's very different from foulbrood. Foulbrood, if we'd have tried any of those experiments with foulbrood, we'd have moved foulbrood in all of those moving, feeding pollen probably, moving honey, yes, moving brood definitely. You move foulbrood, but with chronic bee paralysis, it's not the same. And so for me, I didn't do anything with any of mine, and I've not had it since. Okay, so that's all I can say really.
Okay, thank you. That's really helpful. Now we've got some other questions. I've got Mark F.
Hi, Giles. Yes, Mark. I'm from Wiltshire. I was
Hi,
interested to
Mark.
know, as a fellow virologist, is there any evidence for selection of specific strains of the virus as we, back from the 60s to current strains?
It's a great question, and as part of a project on chronic bee paralysis virus, I managed to get hold of some of the original virus samples from Bailey from the 1960s, and I tried to sequence them, and I tried to inject them into bees to see whether or not I could get any vestigial virus that might be left to replicate, and I couldn't detect anything. They were too far degraded. So when it comes to the old 1960s viruses, I can't really say anything about it. I've got a couple more vials left, and I'm kind of waiting to see if science catches up with dealing with old degraded virus samples. If it does, I'll try them again. Certainly before I retire, I'll try them again. But what we have found is if you look at bees from a colony that has the virus one year and a colony that has the virus the next year, the same colony effectively, then it's almost clonal. It's almost clonal the following year. So you get this carryover of what is almost an identical virus. And if we look at variation across, and this is some work that was done at St Andrews in David Evans's lab, actually there isn't much variation in across England and Scotland and Wales. So it's not highly variable. There are 4 different genotypes. So if you look in the literature, you'll see that there are 4 different lineages. But in our studies, we just found, I think it was lineage B. Okay.
Thank you. We've also got Kirsty who's got a hand up. Kirsty, would you like to ask your question? She needs to unmute.
Yep.
Hi, Kirsty.
Hi.
Hello.
So yeah, last year we had an outbreak of chronic bee paralysis virus in our apiary. To be honest, it was only picked up because one of the people that went around inspecting the hives that year was doing their BBKA module on pests and diseases.
Well, that's good.
We chose to cull the colony because in northwest England there aren't a huge number that have been reported. We then had a talk from Buckfast bees, and they think it's connected to unsymptomatic nosema. Is that something that you've looked at?
I've seen some old references which talk about a correlation between what would have been Nosema apis, not Nosema ceranae like we have mainly now, and chronic bee, as well as Nosema and black queen cell virus. We did look at this in the very early days, I would say when I was in the bee unit in about 2007, 2008, and we couldn't find any link between the two. If we look at our data sets where we have Nosema alongside chronic bee paralysis virus, we don't see any links at all.
But you don't with Nosema ceranae?
No, not with neither of them.
Okay.
Thank you. Rob Cook's got a question.
Thanks, Giles. Really interesting. I suppose the only thing you've said which surprised me a bit was the density issue, that denser colonies are more susceptible, which I can understand when you think about it. When I've had it, and I've got 3 apiaries, and it's only one apiary which has had it, and I've had it for about 3 years now, at different colonies. It always seems to be colonies which are under stress. So last year, I was overwinter with a Nadirid super. That rookie error, I had some drone brood in that Nadirid super, and I didn't get to swap the boxes around soon enough, so I had a load of drones, and that colony collapsed really quickly. And so my where it's occurred to me, it's where the colony hasn't been healthy. Now there's a cause and effect there, I'm sure, but if it hadn't been for CPV, I would have thought the colony was unhealthy anyhow. because too many drones or queen problems or something like that?
I have seen some beekeepers that have had persistent problems that carry a lot of drones in those colonies. And so I'm not surprised. I know that they can catch it. We might have some work coming out later this year which looks at the spread and how well they spread it, in the same way that we've got our pot studies with just workers. So I'm not surprised to hear that about the one which had high levels of drone in it. I am surprised to hear that it tends to be the stressed colonies that get it, because a lot of bee farmers do get it. We saw the professionals. Actually, a lot of their colonies, when I've been round, an awful lot of them are actually in pretty good condition. You know, they tend to keep their bees very well because they're paying their mortgage with what they managed to produce. So I'm quite surprised at that. People more often than not say it's the stronger colonies that get it rather than the weaker colonies that get it. So I've not heard that before.
That's interesting, because my assumption being that it's more or less endemic in colonies, that when colonies were stressed, that it allowed the symptoms to flourish a bit, but obviously not right. Thank you.
Thank you. There's a question on the chat that raises the issue of there was a talk that if you got to chronic bee paralysis, you should take the colony further away, shake all the bees out, and just deal with what came home.
Yes, I've heard that one. So that was originally from a bee farmer who tried it and said it kind of worked for them. I've spoken to a lot of people that have tried it and asked them what the outcomes are, and the colony death from people that have tried it that I've spoken to is about 50-50, which is about what you'd expect if you did nothing. So now we know more about the fact that bees that have the virus and can even have symptoms of the virus, and so they've got very, very high levels of virus, can fly and orientate. And you know, I've yet to see any good evidence that works. And the more we understand about the biology, the more I think it might not work.
Thank you. That's really helpful. I think, do we have any last questions? And if not, I really want to thank you very much for this. It's something that, as your figures show, we've seen more and more of. And you do hear all sorts of ideas about what might work. The more we know about the biology of it, the more likely we are to be able to deal with it. So it's really wonderful to have that sort of explanation that takes us through so that we can understand it. Thank you.
You're very welcome. Nice to see everybody, and thanks for sticking with me.
Thank you very much.
Okay.
Thank you.
Thank you.
Thank you.
Thank you.
Thank you
so much.
Thank you.
Thank you. Great science.
Thank you.
Thank
you. Really helpful. Thank you.
Thank you, Giles.
Many thanks.
Thanks, Giles.
Thank you.
Oh,